is that it's basically a way to look at shotgun sequencing. So you're gonna have 3 segments. There's gonna be like something like that. What you really want to look for is the overlap, and there's gonna be there's gonna be sections of DNA that are identical right here. Then there's gonna be sections of DNA that are identical right here. This equals, so when you connect these, and you can do this on your paper, and you'll probably, you can write it all out if you want. What this will look like is something like this. So this is mimicking the same one right here. Find the second overlap. Oops, sorry, yeah, and then all the way. And this is your full DNA strand. And remember, there's another one that you'll probably have to do it for, because the 3', remember, DNA has 2 sides to it. So you probably have to just solve 2 at once. And I think that'll go, that would still look something like, it'll be a separate set, and you'll be able to do everything just like that again. Okay, but just focus mainly on what I just did right there. Find the regions of overlap here, connect there. Find these regions, connect there. Just like that. Okay. So essentially we also ran a bunch of sequences. So say that here's DNA 1, 2, 3, 4, 5, 6. DNA from 6 different people. Remember, we had asterisks where the DNA was conserved. That means that there's no changes in there. When we had a gap, it means that some people had an A nucleotide here, while others, for example, could have had a, let's do a better one than T, could have had a G nucleotide here, for example. This is an unconserved region. This means that this region can change without harming these people, and that these regions right here, though, here and here, they're conserved. And that means that if they do change, we don't usually see it, because changes in these regions, they usually don't survive. That's why things are usually conserved, is that these regions of DNA are very important to the protein they code for, so you don't want to change them. That'd be bad news. So to visualize that, remember that conserved regions, it's gonna be hard to draw, conserved regions are really tall. So like this A right here, and this, so let's say this G, so there's right here. When they're really tall, remember these web logo things? That means that the taller they are, the more confident that all the sequences you're gonna see are gonna have this sequence. And then if you have all these little guys over here, it could be a C, G, or a T, this region is not conserved. Region, so maybe a little higher frequency for C. I'm losing track. So when you have little regions like that, these are not conserved because the frequency of each one is kind of small and kind of even. But when you have big regions like this that are really clean and big, it means that none of the sequences you analyzed had any changes. These are always what you're gonna see in that sequence, for example. We can go, I think I'm missing one of my colors. And then say that, just big C, big A. Oh, look, like a cat. Anyway, that means that this section of DNA and this one, when they have the big letters, that means it's really conserved. When you have the little letters in these sequence web logos, that means it's not conserved. That means it's variable to change. So keep track of that. So the other thing is that when you're reading DNA, and this is kind of a tough concept, remember that, here, I'll just try this. Okay. So there's a bunch of ways to read DNA that we covered in class, and this is what open reading frames are. Open reading frames are where the protein starts. Let's actually do this just to make it easy. Okay. So the thing is that you could be reading it, there's 2 things that you're gonna have to account for. One is that this could be the 5' to 3' direction when you're reading it. Could also be the 3' to 5', and that's very important. So that's 2 different ways to read this same set of DNA. The next set is it gonna be read for these 3 codons at a time? Just like in this order, remember, because it's 3 and 3. Or is it gonna be read like these three? So it just changes because we know that 3 go at a time. We don't know in what order though. Same thing. See how there's, codons are 3, that means they could be starting in all these different regions, right? So each color represents a different frame that I'm reading things in. So the green frame is if coding starts here. It goes 1, 2, 3, 1, 2, 3, 1, 2, 3. Now, what you can look for is right here is the HEG. I don't know if you're gonna have to memorize that or not, or that's the start codon, but essentially the computers can do that for us and know that this is where the 3 starts. And because an HEG is your first 3, that'll be these ones, this codon, and then whatever's left over here. So remember that there's a lot of different ways to read this, and it's a lot. So a single strand of DNA has 3 different ways you can read the codons, because you can start at one of the 3, if you know what I mean. So if that doesn't make sense, basically you can start with these 3, but yeah, let's do this. You could start with these 3, you could start with these 3, or you could start with these 3. You can see how those are each 3 tiers, and that's gonna be trouble to reach, so which one do you start on? And you have to account for the fact that it could be this direction, or it could be this direction. So that gives you a total of 6 things that you have to be ready for, and we just saw that in lab. Because each codon codes for a different protein, and when you code for different proteins, it counts a lot where you start. And that's why HEG is a big deal. But there's a lot of ways to read that frame, basically. Definitely find a video on this because it can do a lot better than this mess right here. All right, so definitely email me, clarify with me, let me know what I can add or what you guys need.